of how best to use metabolomics in your research, especially if you're. So what I'm going to talk about is actually a story that we have been doing for the past several years, but this is one of our earlier studies. We have actually followed this with many studies down the road. Some of which you'll be listening to in subsequent talks that are going to be ongoing. But this study is just illustrative of how best to use metabolomics in your research, especially if you're doing clinical or biomarker discovery type research. And in general, you heard this morning from a lot of folks, and continue to hear during the course of this symposium, that what are the problems in interpreting the data with metabolomics, and how you need to be cautious, how diet, sample prep, et cetera, play a major role. And you may be wondering, how the heck am I ever going to do a study? So this is for you to assure you that not all studies need to be perfect, but your interpretation of the data need to be cognizant of the facts that actually does what goes well and what does not. So this is our work. My laboratory works on diabetic kidney disease. And so this is the concept that you have been working on in terms of what the idea is and what the workflow and the data interpretation methodology. So the first question that comes in terms of our own research has always been, why do we need biomarkers to begin with? And the answer to that is that the human beings are all quite complex. And it actually came in a very nice words of Dr. Osler, who is thought to be almost the sort of the modern era pioneer of the clinical medicine of the United States. So you can see that he has a Sir before him. So he's not actually, he's one of the people that landed, in the Ellis Island type situation. It actually was in New York for a month. He was quarantined when he came to the US before he was able to allow to practice medicine in the United States. So he actually says a very interesting thing. So variability is a law of life, as no two faces are the same, no two bodies are alike, and no two individuals react alike and behave alike under the abnormal conditions which we know as disease. And this is all the more relevant for human disease than mouse model studies or cell culture work, which are a lot more reproducible, as you know. And so in terms of diabetic complications, so as you know, diabetes is a huge problem. 30% of the people in the United States have a tendency towards diabetes. Roughly about 16% have the disease. But the major reason why people suffer from diabetes is not due to the disease itself. It's obviously a chronic disease. You can take pills or insulin. It's because of the complications. And there are numerous complications. Basically, it affects every single part of the body. And our work is related to the diabetic kidney disease, which is called as nephropathy. So if you think about what the global problem of kidney disease is, roughly, as you know, now there's all the healthcare reform that is going on. 14% of the US GDP is spent on healthcare. About 14% of the federal budget is spent on healthcare. 2% of that entire budget is spent on taking care of patients on dialysis. So this is only 60,000 individuals as opposed to 300 million Americans that are there. So this is a hugely expensive disease. Per capita, probably the most expensive disease in the United States. And so the number one cause of this is diabetes. So roughly 50% of the patients on dialysis in the United States currently are on diabetes. So per patient, this is by far the most expensive disease. So fundamentally, the way we actually define how diabetic kidney disease actually works is by looking at histopathology. Pathologist tells us, if you do a biopsy, what the kidney looks like. This is a normal kidney, which has a nice looking filter, and filters the urine, and you're all happy. But then when you get diabetes, you get this massive deposition of eosin-like material, which essentially leads to scarring and glomerulosclerosis. And this particular thing is essentially what patients with end-stage kidney disease have, and they need dialysis. Now, how we go from here to here, it's not very clear. And in fact, there's varying trajectories of this disease. Now, we know that controlling your blood sugar actually helps the onset of the disease, meaning that you don't get the disease to begin with, but nobody knows about the progression. So people that once you get the disease, controlling blood sugar after that has no benefit in terms of actually the progression part. The degree of protein that you spill in the urine has some role in the disease, but we don't necessarily know how much this actually truly affects the disease process. Lipids, you will actually hear the Farsad of Sharia giving a talk in a couple of days, and mainly on this particular portion of work in the laboratory, and some drugs that actually are known to be important. But again, all of them kind of delays the progression, but they don't necessarily know what exactly is going on. This is one of the reasons we need good biomarkers. So the goal of our study was to determine what are the biomarkers that are associated with early diabetic kidney disease in type 2 diabetes, and to evaluate these associations with progressions. So for this, we actually took an approach, first of all, to ask ourselves a question: can our biological questions be answered with metabolomics? And of course, I won't be standing here if I don't think that's the case, but there is some reason to think metabolites play an important role. When you have end-stage kidney disease, classically what people refer to as uremia, what they're describing is the uremic illness is largely due to the accumulation of organic waste products that are the kidneys unable to excrete. So by far, most of them are going to be metabolites or small molecules, because the kidney is not synthesizing large proteins. Liver is the major reason where proteins are synthesized. The kidney are by far, the excretion products in the urine are going to be small molecules. Now, there have been 160 plasma retention solutes, as people have called it, that are known to accumulate in subjects with kidney failure. And this has been detected mainly by targeted analyses. People have gone in and looked at certain specific tubular excretion products and came up with these list of 160 compounds. And then basically this particular group, over the course of about 2 decades, came up with these findings. And these are the major classification of these so-called middle molecules are. So what are the issues that we need to consider when we are designing this metabolomic study? So the first issue that we wanted to understand is that what would be the study population and study design that we need to think about when looking at this problem. So we came up across with this cohort from the Joslin Clinic from the Harvard Medical School, where one of our long-term collaborators, Andrei Krolevsky, resides. And he has this cohort of patients that have been taken care of for the past 50 years. And they have very systematic data gathering, biorepository, urine, plasma, et cetera. And we divided these subjects into those who progress and those who did not. Now, these definitions are important but not relevant to this topic, is that you have to understand clinically what the indicators for progression are. In this case, it was quite simple for us. Basically, we said anybody that needed dialysis or a kidney transplant progressed. And the non-progressors are people that did not reach those endpoints. So the pros of this group, I mean, the nice aspects of this group was basically that these are very well characterized. There's longitudinal long-term follow-up. These patients come back every year, and they're biorepository every year. And there's data for more than 20 years. So this is an extremely good group of patients to work with. So what are the cons? What are the issues that we need to be thinking about? The problem is that the people that reach all the endpoints of dialysis, there are a lot of people that die before you reach those endpoints. So there's a lot of mortality involved. So not everybody that is followed over the past 20 years actually reaches the endpoint. The numbers are quite small. Even though these people have been followed up for a very long time, when you are looking at metabolomics, when you're looking at large numbers of variables, and you'll hear from the statistical talk from George in the next few days, you lose the power because you're looking at too many variables, and so you need a very large number for you to actually come up with meaningful results. Then there's other confounding variables. There's the age of the patients is different, sex, duration of disease, whether they had high blood pressure, which can also lead to end-stage kidney disease. Their lipids are different, And where they start out in their kidney function is different too. Some people have advanced kidney disease to begin with, and they rapidly progress to end-stage kidney disease. Some people have no kidney disease, and they have been followed for a long time. And the medications they are taking, et cetera. So you have to consider all of these elements when you actually design your study. So then the second question is, what do you analyze? And so in this samples, we said, okay, biosamples. And so what are the things that you have here? You have plasma and urine from the subject. So we decided whether do we do plasma or urine. So the advantages is that both the plasma and urine were collected about 20-some years ago. So we have this very valuable cohort, but baseline samples, you can ask the question, what are the metabolic abnormalities that actually predicted long-term outcomes? But the cons of this was basically the plasma was non-fasting. So as you will hear repeatedly during the course of this symposium, diet has huge implications on metabolism. So for example, if you're looking at glucose or amino acids, after you eat a meal, they are going to be far different than before you ate a meal. So ideally, you want a sample that is fasting, and that is your best sample. But again, we can't go back 20 years and change the way they collected the samples. So this cannot be changed. And the urine, unfortunately, at those days, 20 years ago, they did not know how exactly the urine degradation situation worked, because nobody knew about metabolomics at the time. It was not stored properly. So the question always is that, do you get degradation with storage? Plasma is less of an issue because they were put in -80 right away. So it's very unlikely that the degradation was an issue. But again, you're talking about samples that have 20-some years of storage. So the third question is that, what type of metabolomic experiments do we really want to do? And so here you could do targeted versus untargeted. We chose to do untargeted first, and then we are doing a lot more targeted now. And obvious reasons is that untargeted, you get a broad discovery approach, and then you narrow it down to what you're interested in, and then you can kind of do sort of more reiterative process. So the cons of this approach is basically, again, the small number of subjects. So with untargeted analysis, you get large amounts of data and variables. With small numbers of subjects, and basically you may end up finding nothing because of correcting for all these variables and the multivariable, so the correction factors that you need to introduce, the multiple comparisons. Additionally, for even the clinical outcomes are, the outcomes in terms of age, sex, and all of these things all also need to be corrected for between the patients. So if you do a proper analysis of all of these things, these issues will become a problem. So the third issue is the variability of the analysis itself. So some is technical because you're running all the samples over time. And then the second thing is we talked about the non-fasting state, the diet, and the degradation over time. So all of these issues you need to consider when you think about what type of experiment are you going to do. Now, at the very least, you need to consider it at when you analyze the data to make sure that you at least think about these things. So how did we come up with what we wanted to do? So this first thing we did was something called as CaseControl Cohort study. So what this means is that basically here the case is the people that progress. Control are the people that did not progress. So you want to know what the difference between the case and the control is. What you want to do ideally is that get rid of, basically match factors as much as you can when you select these patients. Now the downside of this case cohort approach is that you're highly selecting your patients based on what you're doing, and that introduces a bias in of itself. So that is the downside of that. But that is the approach we took. So we took roughly about 40 people that are non-progressors and 40 people that actually progressed. And if you look at the most of the things, age, duration of diabetes, body mass index, hemoglobin A1C, which is a measure of how well your blood sugar is controlled, cholesterol, blood pressure, drugs, etc., there's no significant difference. So you eliminate most of the differences up front, at least by clinical characteristics, to kind of make your cases and controls pretty similar. Now, we still could not eliminate all things. The degree of protein in the urine was different between the two, and the estimated GFR, which is a measure of how well the kidneys are working, was basically different. So the people that did not progress started at a little better renal function, and the people that did progress actually had a slightly worse renal function. But you can handle this statistically by correcting for these factors once you do the analysis. So this is one approach that you could take. This is the approach we took. So this is the sort of the untargeted platform that you would have heard about. So we did two different modes here. One is Hillick and the other is a reverse phase. And this is just to tell you that you get tons of data. This is basically retention time over mass. Each point is one metabolite. Essentially, we get numerous data. So they're talking about close to 6,000 or 7,000 points. But not each of those points, we really know what these metabolites are. So I'll come to that in a second. So this is the issue number 4, is basically too many variables, right? So basically, you're looking at outcomes with Untargeted Platform. You're looking at 10,000 features roughly in those 2 different platforms that we talked about. But so you could handle this somewhat in a different way. So what we did was here, we limited only to known and named metabolites. So in other words, all the unknown features or unnamed metabolites were thrown out. Now, there is a downside to this approach. The downside to this approach is essentially that you are actually eliminating data that may be very important. But what we decided was that given our small sample size, we really wanted to know exactly what metabolite we are dealing with. And that actually cut down from 10,000 to 445 compounds. Now, the second thing we did was basically limited compounds detected in at least 80% of each of the groups. So in your control versus the thing. So the reason why we did this was to show basically limited to metabolites that are reproducible and are present at a high number of individuals in one group. So again, this actually cut it down to around 262 compounds. So all the way starting at 10,000, we are down to 262 currently. And then the final issue, the other issue that we want to deal with is basically issues with sample collection. So what is the effect of the prandial state? I was saying that this is actually non-fasting plasma sample. And the question is, how do you handle that? Because you're going to have people that consume meals 2 hours earlier, 3 hours earlier, or overnight fasting. So they were all over the time frame, morning till evening. So we have no idea when their last meal was. So, and then the other issue was the stability with storage. Even though these samples were derived 20 years before, and we think most of them were stored properly, some of those samples have been thawed once or twice. So it was not entirely clear whether those freeze-thawing cycles would have an effect on these compounds. So what we did was, basically, we asked a question. So we took the baseline samples and did other samples from the same individuals a year or 2 down the road from the baseline. So this is, you're talking about year 20 versus year 18. So the idea here is that if you're detecting the same compounds in the same individual 2 years apart, that actually means that these are actually relatively stable compounds and not as much influence with diet, because it's very unlikely the patient exactly consumed 2 hours or 4 hours. This was exactly reproducible 2 years down the road. And this also answers the question of stability part. So this is the sort of the data. So tracking metabolites with individuals over time, what you have is that amino acids, carbohydrates, lipids, nucleotides, and other metabolites, and each point is a different metabolite amongst these classes. And we just came up with an arbitrary number of 0.4 correlation coefficient. You can argue, why not 0.5 or 0.6? I mean, you could come to whatever you want. But basically, it is basically saying that these metabolites that have a high correlation coefficient actually are stable over time and therefore reproducible, are not as much influenced by diet than as the other ones that are actually less correlate with each other and therefore more influenced with diet and maybe not as stable as well. So this is a rank correlation coefficient greater than or equal to 4 between those paired samples was used to distinguish metabolites, persistent or transient concentrations. By this criteria, we came down to 119 of the common metabolites tracked well over time. So here's one way of analyzing the data coming from all the way down to 10,000 to 119 for your final analyses. I'm not saying this is the best way to do it. This is just one way to do it. But as a caveats I pointed out, we will be losing some degree of, you actually expect certain problems with this approach because we threw out a lot of metabolites that could have been very important for disease progression. But just so that we could actually interpret the data more effectively, that is what we did. So these are the metabolites that you actually see. So the common metabolites and the stable metabolites. Some of the common metabolites mean that they're actually found in both analyses of the same individual 2 years down the road. The stable metabolites are those that actually had a very high rank correlation with each other. And then, we also noticed that many of these metabolites, over 119, that were differentially regulated, turned out to be associated with the so-called uremic toxins. Again, that kind of proof of concept for us, this is actually an appropriate way to handle the data. So what are those metabolites? Basically, this is fold difference. The way to look at this data is that these are subjects with lower risk for end-stage kidney disease. These are higher risk for end-stage kidney disease. So the red ones or the blue ones are the ones that are significant. And we are just looking at amino acid levels here, and these are the metabolites that actually turn out to be significant. And what is interesting is that basically this is a univariable analysis. So basically looking at, so this is the way to look at it is that it's just like the same thing as an odds ratio. What an odds ratio is that it tells you what is disease risk for progression. If it is 1, it means that both the controls and the people that progress essentially are the same. So essentially 1 means there is absolutely no increased risk. Less than 1 means it actually is protective. So these metabolites actually are protective in terms of lower in the levels if those actually did not progress. And then the higher the levels, that actually means these sort of positive correlation. Higher the levels means higher the levels of progression. So when you do a univariable analysis, what you see is that most of these amino acid, branch-chain amino acids, et cetera, are actually protective against progression, which is actually an interesting finding because in almost all studies in diabetes actually have found the opposite. So this is actually an interesting finding in of itself, whereas the carnitines, on the other hand, were actually more deleterious because they were actually greater than 1. Once you adjust these data by hemoglobin A1C, which is the glucose control, albumin excretion rate, the amount of protein in the urine, eGFR, which is the kidney function at baseline, all 3 of them adjusted, the data really does not change at all. Meaning that our initial way we had actually put these 2 groups together itself was very, very good. So this is a pretty interesting result. And then this is the other group, the uremic solutes, actually are for more than 50% of the metabolites. And so these are also markedly different. And one of the interesting things we found is that some of these were gut microbiome derived. And of course, this is known for a long time, so I just put in this reference a while ago. But basically, this particular metabolite actually was one of the highest correlations, which actually was a very gut microbiome derived metabolite. So when you do a similar analysis for the uremic solvents, you actually see the same thing. So again, this is all adjusted data and the logistic regression model. And you will hear a lot more about how this type of work you can get in the statistical talks about the caveats of using this type of analysis. And then the final part is basically looking at, so one of the issues with untargeted analysis, as would have been pointed out, is that it's not quantitative. And there are some instances when the untargeted platform does not necessarily correlate when you do a true targeted analysis. So what we said was, do most of these metabolites actually correlate by doing a targeted analysis? Whereas we have an internal standard, we have a quantification, and what we found is that most of the significant metabolites are actually basically be correlated when you did the targeted platform. But a couple of them did not, so these are kind of interesting. So indoxyl sulfate, which is a gut microbiome derived metabolite, did not correlate because in the previous slide you might know that this actually was here, and now there's no change, and some of them did not correlate. So this also tells you the importance of being thinking about untargeted data with a little more caution because of the fact this is not quantitative, whereas this data is a lot more quantitative. So the summary essentially is that we reported that the uremic solutes actually predict very early in kidney disease that progression, and the association was independent from other relevant clinical characteristics. And the absolute, one of the interesting things is that the absolute concentrations of all of these metabolites were within the so-called normal range that has been reported for human studies. So basically all of these were in normal range, but actually at the higher end of the normal range. So even within the normal range, there is this wide variety that actually can predict risk for something. So, and the results of the global metabolomic profiling actually correlated pretty well with the targeted quantitative measurements, except a few I showed. So I will stop there, and thank you for listening, and thank you for staying all this while. So I think all of you need a big applause, I think. All right, thank you. So any questions? All right, truly silenced. Oh, there you go.
For the clinical validation of these biomarkers, how many samples did you require?
So, it depends on what the variance of that particular biomarker is typically. So, if, in this particular study, we get only 40 and 40 samples total. So it's only 80 sample study. But remember that we did the untargeted first and then validated using a targeted platform. But typically, remember, based on the effect size, some of these metabolites, there is a log-fold increase. So you probably will need 5 or 6 to really show differences if there is a 10-fold difference in that particular metabolite. But some metabolites, there's a difference that's actually very small. You're talking about 5–10% difference. Mm-hmm. So for that, you may need to do a larger number for you to find significance. So it really depends on the metabolite that you're interested in. But I would say for a typical clinical study, I would say maybe a 20, 30 samples is a reasonable start in each group. So for you to start your analysis with. In another study that you will hear in a couple of days at the lipidomic platform, we actually started with much smaller numbers. And the validation, we did a much larger number. And by far, the data was pretty correlated. Thank you. Yeah. Yes? So in this particular study, when we did this analysis, we were able to detect about 445 known metabolites. And then, it again depends on the study that you're doing. For the most part, I would say, typically in our hands, in untargeted analysis, we typically get around 300 or so, at least. And so it can vary from study to study. It depends on the instrumentation people use and what other criteria they use to call a metabolite a metabolite. But typically around 300 to 400, it seems about reasonable to me. Right. So we could actually, we have, I would say for the most of these metabolites, we have a targeted platform. Some of them we don't. Off the 445, you mean? Off the 445, what's the percentage? I think roughly about half, we will have clearly a targeted platform for sure. But if we work more on that, we could probably do for all of them. For some of these things, we actually did develop a method for nucleotide analyses for the pseudouridine and uracil metabolites. We actually developed a method in the lab to do that. But you could always develop a method for pretty much any metabolite in the library. Yeah.
So you mentioned one of the downfalls of this type of cohort is the patients that died before getting end-stage renal disease. Right. Have you considered a composite outcome of death or end-stage renal disease?
So those studies are actually ongoing currently. So we just got funded by the NIH to do a larger study in the same population, looking at actually it's a very interesting study. So it's actually both a GWAS component to it and a metabolite component to it. So basically that actually predicts 2 different outcomes. One is cardiovascular outcome, because most of these patients actually die of cardiovascular disease. And the other is a renal outcome. So they have GWAS data for about 4,000 of these subjects. And we have the outcome. So basically, then we could do metabolomic analysis and actually link the GWAS to the metabolite concentration. So those studies are ongoing. So I'll tell you about 5 years from now.
Those results, yeah. Thank you.
5 years from now. We just started, so. All right. Thank you.
No more questions.
Thank you.
Thank you again, and thank you to all speakers of the day.
Right.